So today we will discuss about the topic which is monoclonal antibodies. Basically, the word mono means single, right? And clonal means clone. And antibodies, like we are already familiar with, that antibodies are basically the glycoprotein. These are the glycoprotein, right? Actually, these monoclonal antibodies are produced by the hybridoma technique. These are produced by hybridoma technique. And like in our, if we talk about our own body, our body produces monoclonal, like our body produces monoclonal antibodies plus polyclonal antibodies, both monoclonal and polyclonal, both, right? Actually, there were a group of scientists, let me write, there were G. Winter and his team basically gave the concept, that the antibody can be produced outside the human body. Antibody that is produced outside would be compatible with the human body. That antibody would be compatible with human body. Like this, the antibody which is being produced outside the human body, that would be compatible with the human body. For example, we have anti-tetanus serum, which is basically produced in the body of horse, right? ATS, but it is utilized in the human body in order to treat from the tetanus, right? Tetanus is a bacteria, right? So basically, these monoclonal antibodies can work together. Like they can work single and they can attach to toxins as well, or to drugs, to radiations and all that. Basically, they are used to detect. They are basically used for the diagnosis purpose, like for the pregnancy test, for the hepatitis, right? Now, if we talk about some scientists, like some contribution of scientists, like there were scientists, basically they first produced these monoclonal antibodies. They were named as George Kohler and Caesar Milestone. Basically, if you talk about year, that is 1928, I guess. Oh, sorry, 1925. Oh, sorry, 75. 1975, right? George Kohler and Caesar Milestone and basically Neil Karkach as well. They produced these monoclonal antibodies and they also got the Nobel Prize. Right? They got Nobel Prize on medicine and physiology. Like these can be also asked in the MCQ section as well. There are two more scientists basically which basically implemented these monoclonal antibodies and found the treatment of cancer. Treatment of cancer. Like those scientists were James P. Allison, and there was another scientist known as Tusco Huncho. Right, basically these were the scientists who used these monoclonal antibodies in order to in order to get the treatment of the cancer. Right, basically these monoclonal antibodies are derived from the these are derived from single WBC. These are derived from single WBC, this white blood cell, through genetic engineering, through artificial genetic engineering, through artificial genetic engineering. Right. Basically, these monoclonal antibodies are produced by hybridoma cell culture. Right. These are produced. Basically, it is a whole technique. Hybridoma cell culture. This is basically a technique which is used to make the basically it is used to produce a monoclonal antibodies. Actually, these antibodies can basically join, like they can bind with a specific epitome, right? These antibodies bind with specific epitome. Right, epitome basically means the binding site. This epitome means binding site. Like every antibody bind with a specific epitome. For example, consider that this is a antigen over here. It is an antigen, right? And over here we have the particular sites, site A, site B, site C, and there is a rule that every antigen it basically requires a specific antibodies. Antigen requires specific antibodies, right? Or consider that this is the like it basically requires specific epitome, right? Okay, this is the region, this is one region, this is one region. Let's name this A, B and C. For this part of the antigen, it requires a specific antibody. For example, it is a plasma cell, and we know that plasma cells are basically the WBCs. These are these are white blood cells, right? So basically this plasma cell will divide mitotically and they will produce a lot of plasma cells that will have the specific epitome of this antigen. Same as in this case here, another plasma cell will be there, and there will be replication of a lot of antibodies, and that will only attach to this specific epitome. So by this, like there is the derivation of the antibodies from a single WBC. As you can see over here, this plasma cell was a single WBC, and there is a production of antibodies from a single WBC through genetic engineering. Right now, this is basically this is quite natural. This is already done in our body, but it takes a lot of time, and whenever a disease enters into a body, we don't have that much time. Obviously it will take several days, but our body don't have that much time. For example, you got caught by a snake, and obviously your body will work against that antigen, but it will obviously require a lot of time which your body don't have. So what we will use, you will use hybridoma technique in order to make the monoclonal antibodies outside the body, right? And then you will utilize those monoclonal antibodies. You will inject those monoclonal antibodies into our body so they can work. So this system which I'm talking right now is whole naturally, but this system is also done outside our body through the means of genetic engineering, artificial genetic engineering, right? So it is basically time taking process. So this is how there is production of monoclonal antibodies. One WBC is producing several antibodies, right? But as you can see over here, one is producing for this, one is for this. It means our body produces monoclonal antibodies as well as polyclonal antibodies, right? If we'll combine together them all, so this statement is proved that our body produces monoclonal antibodies plus polyclonal antibodies, right? So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section. I'll be very glad to answer you guys.